Well, welcome back to the the fourth in the series to complete it, and I call it Putting It Together, a year in a plant's life, where I'm going to try and in an hour or an hour and 15, and for some guys that have to leave, that's fine. This will be posted. You can review it. It's fairly comprehensive. It's quite long, and sometimes I talk a little faster than I should, so you may want to go back and look at it again. But I'm going to try and bring everything into perspective in terms of a life cycle of a plant in 1 year. And I'll show you some stuff that, is maybe not directly related, but I think is of interest and an importance for understanding. So let's get going. So this is what it's all about for us on the farm. We're trying to get that grain, whether it be a cereal, I shouldn't call it a grain, that crop, whether it be canola, whether it be peas, whether it be wheat, whatever, into that bin at the end of the year. That's the whole goal, safe in the bin, because that's where the money is. Now, we know that the yield is impacted by a lot of things, and I don't usually use this slide, but I think it's important in this circumstance. You'll notice that most of these things are things out of our control: wind velocity, evaporation, oxygen level, and so on. Then you take a look over at the purples here, and that's all soil pH and rooting depth and nutrients and field locations and crusting and field slope and soil drainage. And we can't do much about these. We don't control the carbon dioxide level. We don't control hail. And when we look at our soil, when you put your seed into the ground, and that's where I'm sort of going to start this whole thing, you've got those things are done. So you should be trying to do something about fixing your land over a long time. If you got pH issues, for example, you got drainage, or you got compaction, because those will haunt you for the rest of your farming career on that land and interfere with rooting depth. So really what you got that you have control on to do all that are these little parts down here: rotation, seed bed firmness, pesticides and timing, your pest management, your swathing dates, your variety, and then a whole bunch around seed. And then tillage, fertilizer application, the stuff we've been talking about. What can you do primarily with your fertility program and your agronomy program is what you have control of on the whole once you're going into the field. So first thing I would say is, if you've got problems, fix them. Things like acidity, fix them. If you've got solanensic soil, you've got some really serious issues. If you've got salinity, you know what? We know that with salinity, if you don't flush out the saline materials out the bottom of it, they just come back and haunt you. In some cases, it's best to just take it out of production. So let's now move on to things you can do something with. So how do we grow a crop? Well, you've already done some of this. You've put some fertilizer down, maybe debanded or broadcasted. If it's elemental sulfur, you don't want to incorporate it. So you've already done that. Spring action, you will have, when spring comes, you'll probably use a burn off if you're minimum zero tillage. And then after that, you're going to seed. So we're going to spend a lot of time with that. Then we're going to get into protection season, where there's going to be some action you can take while you're doing your pesticide applications. Because once you get too far down the pipe, there's not much you can do with macronutrient applications after seeding, because you can't get it into the plants on our broad crops like canola, wheat, and peas, where we can't sideband, we can't really drill in easily. So we're going to use foliar sprays, we're going to stream it, or we can broadcast some things that are mobile, like urea, which we should protect so we don't lose that nitrogen. You can do some work with, say, boron, which is mobile, so you can spread it on any of your anions, which move fairly quickly with water if you get some rain. You can't do much with phosphorus because you can't get it to move. You can't do much with potassium in terms of massive volumes. You can foliar apply and get it into the plant. So then we're going to take a look at what we might be able to do specifically here, foliar nutrients with protein in wheat, because that's always an issue. And one thing I want to mention is, harvesting in trucks. We do a lot of running around. If you think about farming, as I do, it's mostly about going across a field and doing something, but you're going across that field, and every time you go across that field, you're compacting something, and you're compacting it with trucks. And I think that's having a long-term impact. Every field I go into, I find compaction zones 4, 6, 8 inches when I take that shovel into the ground, and at the driveway, it's really compacted. So, maybe it's our time to start thinking about controlled trafficking, at least in a partial way. Because we used to think that zero-till helped remove compaction, but in fact, the data doesn't seem to be the case. And it's that first pass across the field in springtime that, from what I know, gives you the most compaction, and tire pressure is key to reducing that. Anyhow, those are some broad things. And we're going to talk about starting this process here with seeding, and we're going to end up, of course, combining it and taking it into the bin. And the way I look at it, seeding is like this big mass in the cascade. If you don't get that right, these little dribbles down here, which are going to be what we do with our foliar sprays and so on, is not going to save you. This is a huge amount. Most of our nutrients go down with seeding, and especially the immobile ones like phosphorus, potassium, some of the nitrogen. So get this right. Get this right. So let's take a look at the plants we grow. We grow annual plants, monocarpic plants, a life cycle where we flower once and die. And you should know that with annual plants, they are genetically programmed to die. And when they die, like everything, they take as much as they can from their roots, from their stems and their leaves, and move it into the seed as the sink, because that's their goal in life, is to produce some seed. And then mobilize and transfer those nutrients to the seed. And that mobilization is different than when the plant is deficient during the summertime. When the plant is deficient, it moves nitrogen and the potassium and phosphorus and magnesium from lower leaves up into upper leaves. And I'll show you a little bit about that. Not too much on that. But when it senesces and dying, it moves almost everything, other than, I would say, the two least mobile are manganese and calcium, because they just don't move worth a hoot from leaves and shoots and so on up into the head or into the seed pod, whichever, and then start the process over again. And we do that in this mess we call soil. And plants are providing exudates, and the microbes are important in releasing those nutrients and protecting the plants and attacking the plants. And there's physical and chemical and biological processes going on, under which we have hardly any control once we put our nutrients in the ground. Things like phosphorus are tied up in the pool. Microbes are eating them and storing them, and then other microbes are eating them and pooping out the nutrients from them. So there's a huge cycle going on all the time. We get nitrogen released from organic matter breakdown, and we don't have much control on that. Tell you the honest truth, because that's controlled by water and temperature. The plant controls the microbes, is my belief, and we mentioned that in the past. So that's going on as well. And we have pH issues, and you can see here that, low pH affects molybdenum. Low pH allows these micronutrients to be readily available. Most of them are available in this mid zone, but micros are not available here. We know that the pHs affect the yield loss, or we get inefficiency when we get into pHs that are low, and we get into inefficiencies when the pHs are way too high. We can fix this acid one, and there's only one way, apparently, from talking to soils guys, is limestone. If your pH is down below 5, you probably have a pH that ranges between 4 and 6 across that field. We know that from variability now. With SWOT mapping and variable rating, we have got a lot more information than we used to have. So fix it. You can probably variably fix it, because it's not uniform across the field. Fix it slowly, one field at a time. Fix problems when you can and can afford it, and it will make your life a whole lot easier going forward. A little bit on that mobility, because I'm not sure I covered it totally. We know that in soil, these nutrients, these anions, are very mobile. These cations, except for molybdenum as an anion, aren't very mobile because the soil is negatively charged and the positive charge hang on to it. And some of these are really not mobile because these transitional metals like copper, iron, and zinc, they're metals. They're not very soluble. Phosphorus, even though it's an anion, just gets tied up so quickly, so it's not mobile in the soil. In the plant, though, we get a different response. When the plant is short, it can move these nutrients, Np, K, Mag, and likely molybdenum and chloride from the lower leaves to up in the top leaves. If you're getting deficiencies up here, that means you're just short. The plant is not moving them up into that zone. And boron, calcium, copper, iron, magnesium, nickel, and sulfur are not really very mobile in the plant. So you got to feed it to them all the time. And you need to know that. If you're going to put down fertilizers in furrow or side banding, you know that nitrogen in the ammonium form doesn't move very much, so it's converted to nitrate and it's mobile. It moves with the mass flow. Any of those mobile soil nutrients move with water largely, and even sulfates and chlorides can produce salinity because those will move in combination with other nutrients with water flow and wind up in low spots, leaving a salinity. So, you know, I think most farmers know most of that. I like to say that, you know, the plant knows what to do. You hear that every time I speak, just so you know, and we have plants that really know what to do. Look at the gene size of wheat, 107,000, almost 108,000 genes in the whole genome, 101,000 in Napus. Tells you why we grow wheat and canola on the prairies and grow those worldwide. Huge gene pool. Ability to adjust to all things compared to barley, which has a relatively small gene pool, or old rapa, same thing, flax, small, chickpeas. So we have the horsepower to do this, and I frequently just call Napus a great weed, and luckily we have it. And the genetics are really what run that. And we have to do this in one season. And what we have here is we have a growth of biomass, and we have water use here with canola and the same kind of curve with wheat. It is this period here where we grow the source of the nutrients, that's the carbon. We grow the leaves, we grow the seeds, we grow the anthers, the pollen, and we get blooming here and we start producing the seed. We then form the sink. So we can limit production of our crop by either having too small a source or too small a sink. When we get our bumper crops, we get a great source, a lot of built up nutrient here, and we can grow a big sink. And I'll talk a little bit about that with wheat in terms of the sink. And one or the other not being appropriate is going to impact your ability to reach your yield potential. It can be source limited or sink limited or both. If you get a year like last year where we had a pretty good season going until it turned hot, so we had a decent source going, and then, man, the plant had to use it all up just to survive and grow roots, and we had no sink. So we got a poor crop on the whole. And we have to provide those nutrients all season long. And you can see that when you look at this in canola, you can see that there's a tremendous amount of nutrient used, sort of in all categories in building up to flower. There's some used later, but we got to get it there early, and we got to get it used up by the time we hit flowering. And we got to build this big solar panel and roots to be use efficient. We need to grow these leaves. We need to get it photosynthesizing to produce leaves, produce the sugars, and photosynthesis drives everything. And I love these canola plants. I see these now in some of these hybrids where you get a huge root You get massive leaf growth and shoot growth right from the base of the plants. We finally got our seeding down to reasonable amounts of 5 or 6 plants per square foot, so we're not squeezing them. And we're getting huge plants with the ability to grow massive solar panels to collect that solar energy, produce those sugars to then support the growth of the reproductive part, which is what we want. And we take these ingredients and we make everything through photosynthesis. And for many of you that know me, I always say that farmer needs to look after 2 things, photosynthesis and stress. Plant knows what to do with the rest of it. But without that sugar, which is the base of everything, photosynthesis, everything fails or is depressed. And in our climate, which we grow a crop in 100 to 120 days, these monocarpic ones, that's a challenge. That's why we're probably going to have a heck of a time ever reaching our yield potential in this climate. With our soils and the kind of conditions we have, we just don't have enough time in most years to reach, 250 bushel wheat, 300 bushel canola. And when we did the canola challenge, we got 86 bushels after 3 years. That was the best we could do on 50 acres. So it's a challenge, and there are a lot of reasons why we can't do that. And we'll probably never be able to do those kind of things under our situation. I found this as I read, which I think will be interesting to farmers, is when you look at different plants, and I didn't know this, but with wheat, we have about 50% of productivity is related to the leaves, 31 to the stems and the head. But with rye, and some of these hybrid rye are really massive producers, you notice that their stems are really important components of yield production, and not so much the leaves, and maybe that's what gives it an advantage. The stem lasts longer, the nutrients are flowing through, leaves will give up those nutrients as they're required. It's an interesting thought, but every plant is different. Every plant genetically is different as well. And every plant within the field is acting differently. And we know that our roots are key, and most of the roots are in the top 6 inches. 50% of the roots are in the top 6 inches, which is perfect for us, because to grow roots deeper, to go down, 150 centimeters, 50, 60 inches, it costs that plant a lot of energy. So anyhow, that's in a nutshell to start. So here's what we're gonna do. We're gonna look at what you do here. We're going to look at where you have opportunities to take action. You got this pre-plant period, you got planting period, then you got this period, which is about herbicide timing, and then you have this period for action, in-season foliars or broadcasting or whatever you think you can do to help your plants up until about, depending on the crop, we'll talk about that, but before bloom. So you can get nutrients, and I think in many cases, if we're looking at later application of nutrients to support the plant later in life, we might want to think about the timing a little bit more. I think we got to get it into the plant earlier than maybe we think. And once I get through showing you how these plants grows, you can either agree with me or not agree with me. And a lot of times it's logistics and your ability to get it done. But here we go. Let's take a look at this first one, seeding. What do we got for tools? Well, these 2, soil test, tissue test, big ones. This year, I understand soil testing was way up, and we should, because man, oh man, there was a lot of nutrient left in the field. Not enough guys are tissue testing, because if you're going to do any kind of in-season application of anything without a tissue test, let's face it, you're just guessing. And I should tell you, working for Taurus here as a contractor, most of our nutrients are put together based on what the plant likely needs at a specific stage of growth, and to help it with stress management and so on. But basically, they should be utilized with a tissue test, so that you can make sure you're applying the actual right nutrients you require for that plant. You may be just wasting your money if you're putting it down. Most people just put it down hoping and believing that the plant will use what it needs, and that's true. The plant will take in what it needs, store it, use it, or kick it out. But anyhow, I think it's useful. And the last thing that not a lot of people are doing, but I think they should be, is take a look at the nutrient levels in your seed at the end of the day, especially the seed that you're putting into the ground as your next one, the next year's crop. But we use seed treatments, base fertilization, row spacing is important. Plant density, I was going to change that, but I missed it. Crop protection, good. You guys are doing good at crop protection on the whole. You're maybe putting sometimes, I think, down too much when it's bone dry. I don't know why a fungicide is going down, to tell you the honest truth. You look at the triangle for disease management. You can top dress, you can foliar apply, and you're always looking at your field history, your rotation, your yield potential, what's your water, looking at pH. So you've got lots of tools that you use. You don't think of them as tools, but they're there for you, and you use them. So let's get down to seeding. So seed is under a lot of stress. It can be from too deep, too shallow, damaged seeds, too dry, salinity, excessive moisture, cold soil, no soil nutrients, poor seed, fungi and bacteria affecting it. A lot of things working on it. A lot of things working on it. So we test our seed because we want to know what germination is. How are you going to figure out what your plant stand is going to be without germination? We want to know what the disease is. We want to know how vigorous it's going to be. And we want to know, what nutrition we need to apply to this seed to start with. And then we want to get to optimal seeding density. Well, we're making progress there. We're making progress. I mean, the canola guys, I'm using wheat here and I'll talk about canola later, but I'll bounce back and forth. The canola guys are finally getting down to 5 and 7 plants per square foot at last, but they're still seeding at 4 and 5 pounds because the mortality seems to be so high. I'm not sure what the problem is there. I got some ideas, but wow, that seems like a lot of waste of money because those spindly little plants, when they get going, don't produce a great crop. So you're using 1,000 kernel weights now in canola, and you should be using 1,000 kernel weights on everything. Look at germination, vigor, and mortality. And I don't even want to get into canola mortality on the whole because it's a cat's nest. But start working on it because seed is expensive. Percent germination, my comment on that is I think most folks on wheat are estimating too low, thinking 5%, 6%. I think it's higher. And what I would advise farmers and coaches or agronomists to do is actually go and do counts. If you're seeding at 1,000 kernel weights, you know how many plants you should have. And especially if you're in variable rate kind of situation, you should be actually monitoring your levels over a number of years so that you can adjust it over time as you develop familiarity with the quality of your soil on a zonation basis and the ability of plants to germinate in that soil. And you have to do that for a number of years to a number of crops so you get a feel for it. Hopefully you got 40 or 50 for young guys, years or crops in you. And row spacing can make a difference. If you go to a 12-inch row spacing, which many are doing, you start restricting how much you can put into the seed row. And if you think about it, if you're going to 12-inch row spacing, you're targeting, make easy number, 36 plants per square foot. That means you're putting 36 seeds in a foot. That's a third of an inch between seeds. And you're pouring in a fair amount of fertilizer into the seed row, or maybe not in the seed row in the side band, but you're pushing it, and you've got a 3/4 inch opener, or maybe you got a disc opener, which is even tighter. And you can look at this kind of stuff happening here if it's too much. Now, there's a difference between growing wheat and there's growing canola or peas, which are sort of indeterminate kind of plants. And we know that canola fills the spaces. Wheat doesn't. Wheat, you got to grow more seed, more spikes, more seeds, bigger seeds, or a combination of those three to give you your maximum yield. And that, again, I always go back, if you think about source and sink, if you've got a good source to support the growth of those seeds, and I'll go through that in detail here in a minute, and you get them there and then you got a good supply in that source and you built that good sink, you can fill it, you're going to get that combination, and that's what leads to it. because it doesn't fill the space. Makes a head. All you can do is make 1 tiller, 2 tillers, or 3 tillers, and then it makes a head, and the numbers of seeds in the head is variable. And I like to use big seeds. For guys that use their own seed, here's a little trick. Big seeds produce, bigger seminal roots, not necessarily more of them. They have more survival, faster growth, more fertile tillers, and higher yield potentially. So where are the big seeds if you're going to go looking for them? Well, this is where I have a lot of fun. Here's a spikelet, and there's 4 seeds in here. And if you pull those seeds out, you see that these 2 seeds right here are virtually always the same size. This one is a little bit smaller, and then the 4th one is usually quite a bit smaller. You can see it right here, quite a bit. And if you take a look at where those seeds are, they're in the middle of the head. That's where you get the 4s and 3s. And the difference is if you just take a look at it, if you don't get any 4s, because each seed is, like 24, 25%, it's not exactly the same because this little seed is usually smaller, but you can, for easy figuring. So if you lose 1 floret per spikelet, you've had 20, 30% drop in yield. Just that easy. So here are these 2 heads. They're not monstrous heads, and most of the tillers are 2s. So you don't want to have 3 tillers. I mean, if you got really good nutrition, you got good moisture, your first tiller is going to produce, a pretty good head. Your primary head will produce about 60, 65% of your total plant production. And then the first tiller will do most of the rest. And the 3rd tiller or the 2nd tiller, depending on the year, will either produce something or it'll be gobbled up as the plant needs it. And now let's take a look at germination. This is germination. As soon as that radical or the coleoptile cracks the seed, that's germination. This becomes seed development. Here you can see canola just germinating here, and this is ungerminated. So let's take a look at germination. Here you can see this isn't germinated. It's going. This one is germinated and it's past now. This one is already at a point where it's way out and it would be in seedling production. And if you take a look at the seed, this is the first true leaf right here. Virtually as soon as this gets moving, this first true leaf is already there, and this is the coleoptile, which is closed until it reaches the surface. Then it opens, and I'll show you that, and then the first true leaf comes out. Now, how does that happen? Well, we know this is work by presentation by Dr. Chakmak. We know that the embryo has a lot of nutrients in it. This is zinc, and the aileron has a lot of nutrients in it. And those are the 2 living parts of a wheat seed. So here's what happens. The plant takes in water, and the plant is sort of in dormancy and controlled by abscisic acid to a large degree, which has to be broken down, and gibberellin start producing, because once it imbibes about 40% of its total weight, you get fluids start to become available in here. And as I said, the living part are the embryo and this little part here called the aileron right here. That's the only living part. The rest of this is dead tissue. It's starch. And it starts, the DNA and messenger RNA has to be activated, and I'll show you that in the next slide. It starts releasing alpha amylase, which breaks down the starch. Now, starch is stored as either amylose or amylopectins, 2 different forms of starch. Some are bigger molecules and some are smaller molecules, so the plant can use them rapidly or slowly. And proteases also activate beta amylase. Those take the starch, produce glucose. It's fed here into something called the scutellum, which is right here. I'll show you that a little better in a minute. And then a node forms here on the germ, and we get the shoot starting. Now, it just so happens, and I found this, that, and you don't think about it, but this is chloride, and this gray one right here is calcium. And I said, the plant needs all these nutrients. We don't think about what's in amylase when we say amylase, but all of these molecules, along with a bunch of sugars and amino acids, are used everywhere in the plant, even though nobody talks about it. So that's what starts the whole thing off. Now there's sort of a two-stage process to the germination. You get this initial uptake of water, you get solute starting to flow around in it because now it's liquid. The first thing the plant has to do is repair itself. It went into this dormant period and was shut down, so the DNA has to be repaired. The mitochondria has to be repaired. We got to make some new mitochondria. We then have this lag period where the plant is using nutrients and energy to just get itself going again. Then we get this growth curve where we get the DNA synthesized, we get the radical cells elongation, and off we go to push that radical out through the seed coat, through the germ coat. And we know here that there's a tremendous amount of zinc required here, and we got data from Ontario and so on, that zinc is really key to germination. In fact, in Dr. Chakmag's presentation, high seed zinc is in seed acts as a starter zinc fertilizer. Now, Dr. Chakmag did a lot of his work in Turkey, where they've been farming for 3, 4, 5, 6,000 years compared to our 100 years, but and they're really deficient. When you look at the staining also, you see that there's a lot of protein where it's dark blue in those living parts, the zinc and the iron, because these are all key to both the formation of new tissues and the catalyzation of all kinds of enzymes. So here's reality. This is discutellum right here. And these are the starches, of course. And here's the aleuron, this little red line right here, and this is endosperm, it's all starch. So this is already germinating because we've got the embryo axis right here, and this is his coleoptile. Here's the first true leaf, and the root is going to come out right here. The shoot will go down, our root will go down there. So as the alpha amylase breaks this down and feeds it through here, this node forms on the germ, and that's where your new plant comes from. And what's in the different parts of the plant? I found this. Look at this, zinc. See, it says those other things show high levels of zinc in the germ compared to the rest of the plant. Manganese. Why manganese? Well, remember from the very first presentation, manganese is key to breaking down water in photosynthesis. So the plant feeds on the seed until it can photosynthesize. The first thing it has to do is break down water, as well as provide manganese and zinc and phosphorus and sulfur to a degree and iron as scavengers for those free radicals that occur in the seed. There are reactive oxygen species produced there. So the plant with these nutrients right in the embryo is already prepared to deal with the stress and a fair amount of boron, to my surprise, especially in wheat. I expect more boron in dicots than in monocots. But that's all good and it's in your seed, but you don't even know what's in your seed. So we took worked with 2020 Seed Labs in 2020, and we took 100 samples of wheat from across Alberta randomly because they had already been run through germination and vigor tests. So we were trying to see if there was a relationship between nutrients and germination and vigor. Well, what do we find? 100% were low in boron, 54% were low in zinc. Well, the boron didn't bother me much because, everything you look at, it seems to be low in boron. And that's a quandary in itself and another discussion. What bothered me was this. I expected zinc levels to be low in southern Saskatchewan, Durham country, not across Alberta. These were samples from the Peace River right down to the border, a few from Saskatchewan, not many. I've looked at seed samples from southern Saskatchewan, and they were all low in zinc. Guys down there in Durham country are putting zinc down on seed dressings and wherever they can, foliars and so on. I'm surprised, and I think we should be seriously looking at this long term, because we're not putting enough back, and we've been mining it now for about 100 years here on the prairie. So maybe we're just sucking up more than we can put in. One last thing about seeding. The big issue seems to be about fertilizer toxicity. And I do a whole presentation on that with different fertilizers, but for today, we're going to just look at these few plants. And look at the tips. I've mentioned that before, and I used this before. You get seminal roots, and you're going to get variable emergence. And that's what you're going to get. Most of these plants will survive. Some will die. Some will become disease infected, and others will be outcompeted, or they'll produce late, give you all that variability you don't want. Now, I'll talk about only two, and I'm not picking on any particular fertilizer because they're made by lots of people. This is AMS ammonium sulfate, and this is potassium chloride. You probably all recognize those. And the reason I talk about these two is because they got really high salt indexes. In other words, they can be quite salty. And you know what happens with salt. When you get a high concentration of solutes, water moves into them. Water moves into ammonium sulfate and potassium chloride very quickly. And I just want to show you. The other thing is these are just like crystals. They're quite beautiful to look at. They almost look like diamonds. And they're really salty. This one surprised me because it doesn't look like a diamond, but it is salty. So all I did is I took various granules, MAP, and some of our granules here, and I put ammonium sulfate right here, and you can see there's only a tiny amount here, and I always put a big one, a medium one, and a small one, and they're gone in 42 hours. Same with the potassium chloride. They're actually all gone. They've totally dissolved. And here's what's left. This is the part that was left of the potassium chloride, and this was left from the potassium chloride that disappeared. There's a few sort of orange little fibers left there. So the issue to me becomes not that potassium chloride is, well, it is innately potentially toxic to the plant if the concentration is too high. Lots of things are a problem if the concentration is too high. The issue, I think, is that the speed at which the nutrient is released in the soil next to the seed is the problem. It's just too high, too fast for the seedling to handle it, or this root tip to handle it. Anything you can do with some of these high salt index fertilizers is put them a little bit away, even in the side band or mid-row band. A little bit of soil in between really impacts the movement. We know that potassium does not move far. Chloride does move. So some of this could be a chloride issue. Ammonium sulfate, the ammonium moves and the sulfate can move. But it's really, I think, the ammonium that is the issue, that drop in pH that occurs when the ammonium is released, and the speed. So they're not bad. It's just that you got to know where to use them. And I think one last example, even if you take a look at this, is barley, and I just do this for demo, and I've seen it a gazillion times with weeds as well. And this is just MAP, and MAP's a great product in the right place. You take a look at this. Here you put MAP right at the germinating end versus the other end, and there's a big difference. It's just luck of the draw. But once you're putting 36 seeds per linear foot on a 12-inch spacing, and you put fertilizer into that seed row, you are increasing the chances of that granule being near the germ end in the cereal, or near the emergence of that little radicle from a canola plant right next to the seed. It's just that easy. It's just sheer random luck. So many canola growers are now making a decision not to put anything in the seed row, just because, to reduce that risk. I think there's no problem with putting potassium or phosphorus in small amounts in a liquid formulation or in the side band. Just think about where you're putting it. There are all kinds of rules here, which you can find in any government publication, about the amounts you can put up. Say this is just phosphorus, and the combination of potassium and phosphorus, because both of them can have some issues, because the potassium isn't pure by itself. You got some ammonia in there as well. And temperature is important. The deeper you go and the lower the temperature, the longer it takes to emerge. So some years you win by seeding as absolutely early as possible, and it warms up. Of course, the soil doesn't stay at that constant temperature, but the deeper you go, the longer it is. So there can be a lot of repercussions of it. If you miss that early period and you think you're late, you know what? The plant will catch up because it'll germinate a whole lot quicker as long as you got moisture there to sustain it. Now let's take a look at germination. This is germination just quickly so you guys can see how your plants come up. This is the coleoptile coming up. Tip opens up and out comes the seed. It's got 5 seminal roots. And then after it gets going, it produces crown roots. And here's the tiller coming off. The seminal roots go down a long way, and then the crown roots come off a little bit broader. By stage 31, and I use this stage because this was where we were looking at manipulator, and this means that this is the seed head, and it's 1 centimeter above the soil surface. So there's your first node. The plant grows from the bottom up. It's got about 10 or 12 nodes down below here, and then it telescopes them up. The seed head is always at the top, so it grows from the bottom up. It's just the way it is. And I did this because we were in the beginning with the use of manipulator, we were going at this early stage. And then here's what happened. I took plots that the company had here north of town near Morinville, and I took the primaries, the first tiller and second tiller from the non-treated and the treated, and it sure worked every time I've looked at it. It's a reduced height. So by this stage already, even before the seed head is above the ground, is formed right at the ground level or just below, this is the maximum number of seeds that the plant will ever have. So if you look at them closely under the microscope, you can see this is one spikelet here. There's another spikelet. And here are the glooms, and here are the seeds forming early. So you can see how tiny they are, and the meristem that is growing here, these tiny little seeds. And from now on, we got to protect what is there. The plant generally has the ability to produce about 7 or 8 or maybe even more seeds per spikelet, but usually by the end of the day, it kills off a big bunch of them. So we have potential. So it keeps on moving on, and then we got 3 nodes here, and then this is going to be the seed head moving up, just keeps on going. So we talked about zinc, we talked about manganese, we talked about phosphorus, and we talked about not so much about boron. Boron is involved in cell wall structures and in pollen tube growth, and I'll talk about that a bit. We talk about calcium because we're always looking at base saturation of calcium in that 70% range, because calcium is key for building cells. So is phosphorus, so is nitrogen. Potassium activates and moves things around. So not that I promote our products, but I just want to use as an example some of those. I should admit I do promote our products because I think they're really good. But it's more important to look at when we build a nutrient package, and this would be going on seam active prime, because we know that seeds are not fully balanced. We've known that for a long time. It depends on what the mother plant had for nutrients, and it's never exactly balanced. So there's always a little bit something missing. So we know that phosphorus is key and is not mobile in the soil when it's cold. We know that potash isn't either. So we put a lot of phosphorus because ATP is key, and we're going to build those early cells. We use a little bit of boron for cell wall structure. We use these because they're involved in enzymes. Zinc is in all the 6 groups of enzymes in plants. It's the only one that goes across or involved in some of them. Probably somewhere between 12 and 1,500 enzymes contain zinc, because these are all catalysts for reactions. Herbicide timing comes along, but I'll go through it. Here's your plant. Here's your coleoptile. The first true leaf comes up. Notice that it's blunt. After that, all the leaves are pointed in wheat, so you can easily tell one from the other. Here's the intercrown node, so the crown root will be here, and the seed head, I'm not sure where this stage this is, but it's formed right here in this crown. That's where the seed head is formed, because there's nothing growing here. This is the seed, this is the intercrown node, so the seed head can only be formed here. Here we're moving to 2-leaf stage. Here we're at the 3-leaf stage, and then we're going into tillers. Here's 1 tiller, 2 tiller, and you say, well, here's a tiller. Well, it is a tiller, but it's a different tiller. This is called a coleoptile tiller. These are actually different, and I'll show you close up. So here's the coleoptile tiller, which is not coming from here, and I'll show you that in a minute. It's coming from down here where the seeds form, and here's the intercrown node. This is a short one here, and then here's your crown root. So here's your crown node right here, and it's going to put on another set of more crown roots here. And here's your coleoptile tiller. You see it starts way down here and comes up. Not sure why that happens, but you know, when you're looking at your plants, you'll be able to dig them up and find those coleoptile tillers. And I think they'll still produce a tiller, but I sometimes think it's due to not enough plant density, and therefore the plant sees more space, or for some reason it stimulates to produce that coleoptile tiller. Now here's a typical tiller, and you can see the tiller starts here at the crown node, and there it goes. So here's the coleoptile tiller. So this one's got two tillers and the primary shoot. And you can tell the tillers from the primary shoot because they have their own coleoptile here that protects it. It's called a profil. And it's got this nice green vein when it's young, so you can stage it better. So if we're going to be going in at herbicide timing, and the reason we developed this product build to go in with at herbicide timing is because herbicides themselves provide some stress, and to deal with the stress and the possibility of nutrient deficiency early in the plant life, and we can't put on a lot of product, either broadcast or so on, macronutrients. We ramp up the phosphorus, the potassium, not so much the nitrogen, and then notice again the zinc. And when in build, we also put in a little bit of moly, which doesn't need a whole lot of moly, to help with nitrogen reductase and abscisic acid formation, and a couple of molecules that are used there. And we can use build combined with most herbicides. And if you go to the website, you can check and see which herbicides it works with and safe with, because you should always look into that. So as I said, a farmer should be taking a sample at about the 4 or 5 leaf stage to see what the nutrient levels are, because if you get behind the curve too far at that early stage, it's hard to catch up. So you can do a tissue test, take the whole plant, get a tissue test, but generally we provide this kind of a nutrient package, and many suppliers do, and they can go on wheat, canola, soybeans, and so on. It's just a general use, a support treatment to deal with deficiencies and stress. You'll notice that from my stress presentation, manganese, zinc, we don't put copper in because copper doesn't play well in some of the formulations. There's plenty of iron in most of our soils, and we have these to go along for that stress management. We don't add any sulfurs, but most guys are putting in broadcast sulfur, and you can put sulfate on the soil surface and with water wash it into the soil. So fungicide timing, either pre or post harvest. Not so much post-harvest, but pre-harvest. One thing I also should let you know is that the kernel number is established about 20 to 30 days before flowering to 10 days after. And the reason is that that's the final stage where the plant is hormonally and nutritionally deciding how many seeds it can support based on the amount of photosynthesis and the amount of sugar available there and the amount of nutrients there. So you want to go into that stage loaded up as best you can. Just that easy. So let's take a look at how this is working. Head emergence. You can see the head is just popping out here. That's just stage 53. And then as I just summarized everything here, you can see what happens is this head slowly comes out as this last part of the plant here to the first node, the peduncle pushes it up, pushes it up, pushes it up, pushes it up, because it's the flag leaf, pushes it up. And that pushing up takes about 5 days. It pushes up anywhere from 1 inch to 1.5 inches a day, and it will typically then pollinate. Now, I should put a caveat that for guys that are looking at fusarium head blight, and I developed this for timing on fusarium head blight, the hotter it is, the faster it goes. So on a nice, cool, high moisture year, it'll take 5 days to pollinate and start pollinating because it's pushing this peduncle up. It's got good nutrition, good water. But if it's 35 degrees and there's no water, it'll pollinate within freaking day, 2 days of just popping out of the head. So you gotta take that into account if you're looking at timing a fusarium here in the first anther kind of stage popping out. That tells you that that's pollinated. This floret's pollinated, this floret's pollinated, and that's just stage 60, 61 is starting, and then it continues from there. And you should be putting on nutrients early, and then let me just go through this, and then we'll talk a little bit about going in with something, another product we have called Grain Fill to help with filling, which you can go to our website and check in. But I'll talk more specifically about using nitrogen as a, because I get asked so darn many times about it, nitrogen in the form of either UAN application or what we call is dissolved urea, where it's put into solution and sprayed on to drive up protein. But let's take a look based on Marshner's book here and so on in terms of what happens in that reproductive versus vegetative stage. I often talk about we're growing the seeds and so on, and I'll show you this more in a minute. But when we go reproductive, everything rises. Boron goes way up, zinc goes way up because there's a lot of activity. You can see that these transition metals and these metals are really ramped up because they're catalysts in all kinds of reactions. So we have to have them in the plant, and the calcium level goes sky high because of the stigma, secretions in the stigma and the pollen tube growth. So they're involved in things like pollen and lignin production and auxins and hormones that are in there. So we get this shift in movement towards a big increase of uptake or movement from the bottom of the plant as best it can into the seed through the xylem. So here we're going into pollination. You can see that here are the anthers, the green ones, and when they start turning yellow, they start producing pollen, and here's the stigma. So the pollen will land on here and it'll go down, and here's the embryo with the seed down there. So here we have it. Here's the pollen. You can see it's stuck to here, and all you need is one pollen tube to make it down to here to pollinate wheat. When you have canola, you need a pollen tube to grow from one pollen grain down to every seed to pollinate it. So you can see it spreads out. It's very shiny, very sticky with all kinds of secretions, because when the pollen germinates, it grows a single cell, the pollen tube, which grows down here, and it has to grow all the way down here, and it has to feed. So you take a look at it up close, you can see that all of these pollen grains are here, and the plant is made with these all these little fringe parts of it that will catch the pollen, just like in corn as well, and it'll put a pollen tube, and it has to feed all the way down into the seed. So the plant loads up all this tissue with nutrients in support of that. So that's what gives you the big buildup going into pollination, because the plant has to feed the pollen. So here's what happens. It's just a simple diagram. The pollen grain germinates, and there's a hole. It pops open here. There's little ports. It grows out, and on the way it splits. The actual nucleus produces 2 sperm cells. Okay, and then it's got a tube nucleus as well for growing that. And this is simple. It enters the ovule through something called the micropyle, and that's controlled by, we think, hormones. Goes in there, the tip breaks open, and the sperm are put into here to fertilize the egg, and the other part is fertilize the part that produces the endosperm. So there's double fertilization takes place. Now here's the key thing. Remember, it says calcium goes sky high. Well, the calcium is used in the growth of the pollen tube because it's got to support the structure of the pollen tube that's chewing its way through the style down to the seed. So the yellow, the pink represents the concentration of calcium. Here's the vacuole, which is back, and it's plugged behind it. So the vacuole is coming in to store nutrients that are being absorbed through the tissue of the pollen tube, and it's feeding it here, and it's just feeding as it goes through the soil. And you can see there's reactive oxygen species here that the plant is dealing with all the nutrients in there. So the calcium is signaling, and I'm sure it's involved in finding its way into the micropyle, and that's why the levels are high along with boron in terms of supporting pollination. There's also high levels of calcium at the root tip, and there's also a pH shift, which also helps guide the pollen tube. So let's go to ripening. Now here's the trick, and I'll just try to explain it. If you want to get your protein levels up, you've got to do an analysis of your flag leaf, probably just before the head starts emerging. I would do it actually in the boot when they're fully leafed out, fully unfolded. If you get about 4.7% nitrogen in that, you've probably got 13.5% protein locked in. If not, you can apply nitrogen, get it into the flag leaf, and get it into the seed, because the plant will pull everything out of that flag leaf before it's done. If you miss that window, and I'll show you photographs of this in a minute, if you miss that window, the next time to do that is after pollination. Now, I want to just make a point here about that source sink issue. When the plant is building the seed, it has to build the storage for the starch, which are called amyloplasts right here. These plastids here, the chloroplast is one type, the chromoplast gives you color, and there's all kinds, but it has to build the amyloplast inside the seed. If it doesn't have the nutrients and carbon to build the amyloplast for storage, you haven't got a big sink. If it's short, then if you don't have a big sink, it limits the amount that you can store into that seed immediately. That's why nutrition going into a seed head formation, and seed formation is key. And then filling it is another matter. So here we go. If you miss the flag leaf, your next stage is right after pollination and what I call the water ripe stage. In other words, right after pollination, it's just a bundle of juice. The plant is moving stuff into here to build the seed. It's not differentiated into anything for a while. If you put on nitrogen, it will get into the seed at that point in time and be incorporated into the protein. If you go late, you'll get some in there, but it'll be mostly not well absorbed, and the bakers don't really like that. So you want to go about a week after pollination. Well, I would say about 5-6 days after complete pollination to get that nitrogen into this seed before it starts differentiating, because now it's going to stop. By the time it's this side, it's not pulling much anymore into the seed. It's now getting ready to sniff. So here's a closer look at this. You see this is pollinated. You can still see the stigma here with the pollen on it. So at this point in time, look at it. It's just tiny, tiny. So this is like a day after it's been pollinated. As it grows, and you can see here's still the remnants of the stigma. You can see that this spikelet is still not formed a whole lot in there, and it will form protein for you. And then as it grows, you can see it starts forming more and more tissue to a point where you have this. And now at this point in time, it's not pulling a whole lot into there. So you're not going to change the protein at that late stage, in my estimation. And then you get this, and now your seed is senesced, plant dies, just like the cycle of life. Now there's one question that comes up: does the plant die from the top down or from the bottom up? Well, I say from the bottom up. And there's something called the stay green gene, which they tried, and what happens is it stays green, but it doesn't move stuff into the seed. Here's the transgenic. So you take a look: grain protein 13.25, 19, zinc 5.2 versus 82, 37 versus 60. These are annual plants. They're programmed to die when they're born. And it starts by extracting nutrients from the roots, from the leaves, because it needs the stem to move them up into the head. And it takes all it can, including a bunch of micronutrients at small traces, because if it didn't, we'd have more deficiencies in the seed. So that's my story on wheat. I covered a lot of detail in that, but the canola will go faster. Here's canola germinating. You can see root hairs develop very quickly. Lateral root hairs up in the top zone also, and there they go. Great weed. One thing is, here's the cotyledon. Right at the base of this is the growing point. This is where, if it freezes, it's dead, because the cotyledon is all out of the ground. There's nothing left in the ground but roots, so it will not regerminate. There's a shot of it. So this is going to be the next new leaf, and this is going to produce the next one after that, and so on and so on. So that's where it is, right here at the base, down and deep in there. The cotyledons come up. We start forming leaves. If you have a shortage of nutrients, you want to get it on before the 5-leaf stage. And canola, 1, 2, 3, 4, 5. Here's 4-leaf, 1, 2, 3, 4. The seeds are already forming here. You want to get on your fertility now, herbicide time or before, okay? And whatever, put it on then. We get inflorescence forming. As soon as you see that inflorescence forming that primary bud, everything inside each one of those little buds has got the seeds and the anthers and everything. So that curve of growth that we're looking at starts way back then in terms of forming those seeds and getting them ready for pollination. It's not just leaves, it's all doing that. If you take that bud and you open it, here you can see here are the anthers. They're green, they'll turn yellow, and this is the stigma where the seeds are. If you cut it open, they already got the seeds right there. They're already formed that early. So if you start stressing your plant out, if you start cutting back on nutrition, you're going to have poorly formed seeds. You may have some blanks, whatever. As the plant moves up and stretches out, it'll put out at the base of every leaf, there's a node. So the plant will put auxiliary buds at all of these nodes if there's enough nutrition, because it's at the node where the plant can transfer nutrients from the stem into the shoot, except for the primary one. And then as this elongates, all of these flowers are stretched out across that stem. There you can see they're going. All of these are formed, and as they grow, they just get bigger. But they're already formed, even these tiny little guys here. And you can see this one is aborted already. For whatever reason, don't know. If you look inside, here we go, take another bud as it gets older. You can see that, of course, when you get to yellow bud stage here, it's just about ready to pollinate. So you already have the ovules well developed along with the stigma. You can see the stigma is right here, and the seeds are already formed. The pollen is starting to yellow. As it moves, we get that stretching out continuing, and we get buds up at the top part all the way. Now, what happened last year? Well, this is what happened. We were just getting into flower when it turned hot, and this is what happened to canola. It was just that easy. Just didn't pollinate. Knocked off all the. So when you get that, it ain't Lagus bugs. These are Lagus bugs chewing on a few buds here. You know, you can see that mouth part. Lagus bug will take one here, one there, whatever, but not when you get 4 or 5 in a series. That's a weather event. And here we got yellow bud. You can see this has been fed on by Lagus bug, because where they defecate, you can tell that it's been fed on. Continues to grow, and here we go. And the photosynthetic, threw this in here because I found that the photosynthetic capabilities, I got this from Phil Thomas years ago, changes as the plant matures, and this is sort of GS60, which is just the beginning of flower, and up until then we got, you know, the leaves are producing a lot of nutrient for the plant, or photosynthetic stuff. The stems not so much, and of course the pods nothing, because they're small. And as it goes to 50% bloom, the stems now become more important, because now the leaves are giving up all that nutrient like crazy in order to feed those buds and flowers and seeds that are growing. And the time we get way on, it's basically the pods that are left green. The stems still have a mulch, but the leaves have already been sucked out, because you know that canola drops all its leaves. As soon as it turns yellow and lyses all of the cells and pulls all the nitrogen, phosphorus, magnesium, and whatever it can out of those leaves, it drops them off. So that shifts over time. So I don't worry about those dying leaves in the canola plant as it's approaching maturity or even pollination. Flowering, GS60. We don't, we go into the flowers there. Anthers are opening up. This one you can see is not ready to go yet because they're not yellow. They start yellowing. You can see all the pollen inside. They open up and all that pollen's dry, goes on to the stigma. You can see right here, this pollen here is put out a tube. And here are all the pollen tubes here. All these pollen tubes here, these lines are pollen tubes from the pollen on this tip. The tip is really luscious. So you can see right here, it's just covered with all these droplets of nutrient waiting for the germinating seed to come along, and then down it grows through that stigma down to each particular seed. You can see this is already finished pollinating. I throw this in simply because you see this all the time. Rare amounts. This is what causes it, this little thrips. There's some scraping on here real early, and that causes distortion, which twists it. It's nothing to worry about. And that just continues up. And of course, the bottom, I should point out this. The primary bud is the first one out, and it's the bottom florets that produce the biggest pods. Reason the first ones pollinated produce the biggest pods and the most seed, because they have access to the most nutrient first. By the time you get to the very few last seeds that are there, they can be pepper if you get dry. They're pepper because there's not enough nutrient to feed them all, so it just aborts those. So a product we would use to support flowering at, say, the sclerotinia stage would be something like this. This is our active flower. Phosphorus, a lot of potash, potassium in there, because we find consistently that late in the growing cycle, canola plants seem to be low in potassium. That's probably because they got most of the roots near the surface. It's usually dry. There isn't a lot of mobility of potassium. They maybe pulled some out of the leaves, and it's short. So we supplemented that. The boron is there for those reasons in pollen tube growth. Copper, iron, manganese, zinc. Again, if you do some tissue testing, and I like to do actually the buds themselves, see what the levels are, you can make a decision whether you need to apply something to canola at that 5 to 30% bloom. This can go on canola, can go on flax, and so on. And you should go to our website and look at all the products we have that are foliar and compare them to other products that you're using. Lastly, peas. Here are peas. They grow pretty thick. They're somewhat indeterminate bloomers. They bloom like canola over a period of time, and that's important, because if you look at a plant, let's go back. If you look at wheat, a wheat head will pollinate all the seeds if the weather is really consistent in about 5 days in the spike. It starts in the middle, it goes up and down, and then there's the next row and the next row and the next row. When you look at something like peas or canola, which are somewhat indeterminate, and you've got a long period of pollination that occurs, you'll probably have the initial flowers pollinated, get access to the first supply of nutrients. So if you go in at about early bloom, and that sort of fits with when you would be putting on fungicides, that's a really good time, because you're going to now be putting on nutrients to support all those florets and flowers that are going to be coming on stream a little bit later in the cycle. So you supplement it then, and I'm going to show you how that works. Now, a lot of guys are new in peas, and I put this in there just for curiosity. You can find it on most websites. You're looking at about 7 to 9 plants per square foot. You can seed it early at 5 degrees temperature. You can seed it fairly deep, anywhere from 1.2 to 3.2 inches, 1 inch to 2 inches typical. They can go in deep to moisture, and I'll show you why. Low mortality, basically, but they are disease susceptible. You want to go with early weed control because it sort of gets going a little sluggishly, and you don't want to damage it too much later in the season. And I should say that you know, there's specific nutrients, and we have one called VPR, which is used specifically for herbicides like Viper. So if you're looking at spraying peas, and I should tell you, I don't know nothing about herbicides, so you shouldn't take anything I say about herbicides too seriously without checking our website, because I made a decision long ago not to get involved in herbicides. And seedbed desiccation is often a disaster for peas. It takes a lot of nutrient to moisturize that big seed. So you want to make sure you got moisture, and you don't want it to stop halfway through, because that can be a problem. One thing, and I mentioned this in my nodulation a little bit, is the plant is lazy. So if you have too much nitrogen fertilizer there early, you won't get a lot of nodulation. But you do need some, like 20 pounds or so, to support, you know, probably, I made a mistake. You need about 20 pounds. We'll set it back, but you need about 10 or 15 pounds to just build that plant. And this is typical growth. So, you know, all of our products, you can see that the potassium drops off really, really rapidly after pollination. That's common in most plants. You can see that nitrogen use in peas, the higher the protein, the more nitrogen you're going to need. It's just that simple. Well, let's look at germination, that aspect of it, because it's kind of cool. So here's your pea, and there's something called the plumule, which I'll show you in the hypocotyl. So here's typical, and people have trouble staging this. And you show, I mean, you get one scale bud and sometimes two, and then you get your first leaf, which got a tendril there, is typically the way I look at it. Now, there's two kinds of emergence. There's the epicotyl type, which extends, and the cotyledons stay in the ground. So that's peas, lentils, chickpeas. Soybeans don't. They come out. So the importance of that is that these, if they freeze, kill off that, they will regerminate, or if it freezes off, you'll not get a branch even coming off here, because the growing point is down here at the seed. If a soybean freezes that cotyledon off, she's done, just like canola. That's called epigeal extension. So just so you know, that's why we can go in early with peas and some of these other ones. I don't know much about lentils or chickpeas, but with peas, we can go in early. With soybeans, you seed late. You don't want to get frost because it won't recover from frost. Yeah, oh, this moved on me. Okay, seed loss is a problem from handling, and here's why. Take a look at a seed. This is just a seed, and you see here's where water enters to imbibe the seed, and this is actually the plumule and the new shoot. So it's just sitting on the surface, and the coating is thin. So if you're handling it badly and you crack that, this is sort of equivalent to the germ on wheat. This will desiccate and you won't get germination. And it's this that actually forms the shoot, and I'll show you how that happens. So here's the seed, and you can see here's the plumule. So the plumule is that you can see it gathers moisture in germination, and it provides some turgor pressure to get things moving. And then, as I showed you in the wheat, that the plant can restructure its mitochondria and all its hormones in there. So it pushes it up, and you can see here that this plumule is attached to each half of the cotyledon to form the plumule. And you'll notice this right here. Well, I'll show you that. This grows up. Sorry, this becomes the plumule. This becomes the root. and it grows on the top. I'll go back to show you that. This is really cool. This just pops up and becomes the new shoot. And here it comes out of the ground. So we have this, and you can see that peas have a pretty good root system, even real early, a lot of lateral roots up here. And then we got that 1 scale node, 2 scale node. And here we have 2 scale nodes. So this would be at the 3 node stage as far as I'm concerned, and same with this one. You can see there's quite frequently 2 of these little things that don't produce nothing. And there's the clam leaf, and within the clam leaf, so when you look at this, here you can take a look at, within that, there's at least 3 or 4 more clam leaves. See, so here's one tendril, here's another tendril, here's another tendril, and here's the growing point right here. So this thing is producing all of these within that clam leaf, and then it releases one with the tendril, and then another one will come out here. So whenever you look at that clam leaf, there isn't just one new leaf coming out. There are 3 or 4 of them already being formed in a microscopic level, and this is all the growing point that forms everything going forward. And eventually you get the plant growing up, you get flowers start forming. Now here's the point I want to make. If you take a look at 2 or 3 of the flowers that are on each I don't know whether it would be called, I guess, the peduncle of the plant, and you look at it and you cut into, you can see that this one is more advanced than this one. You can see the stigma is much bigger. The pollen is forming and it's yellow, whereas this one isn't. So each one of those 3 is at a different stage. So after you see the first 1 or 2 flowers on it, you got all the rest of them that are going to be coming are going to be at various stages, and that's where you want to get the nutrient to. So you take a look at it more. This is even further advanced. You can see the pollen is quite orange in peas. It's quite orange. And here you can see that the pod is already formed with the stigma here, and you can see that this is the tip of the stigma, and it's got these hairs to catch the pollen. The pollen, the anthers start growing out here, and poof, releases it, and there you got pollination. So now the plant will grow all of these pollen tubes down to every one of the seeds, just like this. Stigma, it's got to feed that pollen, and one has to grow down here. So when we get stress on the plant and it can't grow that pollen tube, or the pollen tube busts, another one may grow down. But once the pollen tube hits there, the plant closes it off, the micropyle, to the next one, and it won't repollinate, so to speak. And generally, that's what happens. So you need a lot of pollen, and you don't want a whole lot of stress going on during bloom, early bloom, and pollination. And then you get your seeds forming, and if you don't get good pollination, you get these blanks. And a lot of people blame that on boron. Otherwise, you get really nice seeds, and you get these nice big pods, and you can see here there's 3 of them on this one stem. And the bottom ones will have bigger pods, and if you don't fertilize later on to keep the nutrient level there, or if you don't get water, I mean, water is really key. I forgot to mention that in wheat, you want about 1/3 of your water coming down after pollination. And a lot of times we don't get much of it. So if we're already dry going into it, the plant cannot fill those seeds very adequately. And the same goes through with the big seeds here. And for soybeans, we have, or for soybeans, peas, and lentils, a product called PodFill is there, and we provide that with a lot of potassium for movement of nutrients, phosphorus, some nitrogen, and some boron for cell wall structure development as an aid to support pollination. And it seems to give a lot of nectar production in pollinating plants and enhance bee visitation. And that's it. And that's it, except for this. We go through the season and we're going like mad. We're going like this jet boat. We got hardly anything in the water, and we're doing 100 miles an hour just to get to this point to get it done. And then when we get in the combine, we even go faster. So one of the things that we do is we know you want to go faster. So we've formed some relationship with a company called Duckfoot, which are attachments that go on the spikes here on your header. to help move that through. And we have a way to monitor, a remote monitoring that you drop a pen, and we got a cleaner that allows you to clean your grain, and a map and a scale, so we can help reduce the loss. It's a pity to grow that crop and then lose it at the combine. So we try to provide nutrients for the whole process of growing, along with granulars, which I didn't talk about, but those go up front, along with other granules. And everybody's in a competitive business. We try to use low salt index ones and provide season-long release of products like a Crystal Green, like a Sulfur Plus, and so on. But now we want to help farmers make and do a better job of harvesting their crop. That's the end of it for me. This is the end of the series for the year. We may do some more in detail. These are broad, and sometimes I talk too darn fast because I got to get through this in a reasonable amount of time, and I want to give you as much information as possible. Go back and look at these. I try and provide principles that are more than, do this, do this. I try to look at understanding as best I can and give you what I have access to knowledge in my photographs to help you understand how these plants grows. Thanks a lot, and I hope that was helpful. Good luck this season.